Okay, cool. So my name is April Sagan. I'm a postdoc working with Hatice Osman-Bayoglu at the University of Pittsburgh. Maybe I'll take this off so I can be here more clearly. Yeah, so I'm going to be talking about my work on inferring transcription factor activities using spatial transcriptomics data, and doing that in a way that uses these spatial features, and also what questions can we answer using spatial transcriptomics. All right, so in general in our lab, we're interested in these signaling pathways and transcriptional programs. And the spatial context that surrounds a cell has this cascading effect that's going to affect transcription factors, controlling ultimately the identity of a cell and its function. So, I mean, the last two talks were also on spatial transcriptomics, so I don't think I have to go into this too much. Specifically in this work, as opposed to the previous talk, where I was using something like Merfish, we're using something like Visium, which has the upside of being whole transcriptome, as opposed to a targeted panel. Right, so kind of the interesting thing about this, I think, is as opposed to single cell data, I could say, sorry, I'm not controlling that. I think this is from a recording. I don't know how to. Sorry. I was playing my recording from when I recorded the talk. Anyways, right, so looking at these two genes here, CRIS3 and CXCL14, if I just had single cell data, I might be able to say, right, CRIS3 is kind of specific to one cell type. It varies in a few different ones. So here, I can actually say all that variance pretty much comes from the spatial location. I can isolate it to one location in the sample. A couple caveats are with spatial transcriptomics data. That laser pointer is not going away. Anyways, we have these groups of cells and spots. There could be 1 to 10 different cells of different cell types. And also we have like really sparse data, even compared to single cell data. So that kind of gets noisy and hard to work with, so we'll have to address those challenges. Right, so I'm going to go over the entire methodology before getting into each in particular part in more detail. Starting with the kind of data that we work with, we have spatial transcriptomics data. So that's gene expression, it's spatial coordinates, and we also have images, right? We can align these gene expression data to these images and get like an image of each spot where the genes are originating. Right, so in this project, I'm working with a list of transcription factors and their target genes. Right, it would be kind of way too hard to infer that from the spatial transcriptomics data, given how noisy it could be. And then we'll use at one point a single cell RNA-seq data set as a reference, just to get like the an estimated gene expression profile of different cell types. The kind of bulk of this is the rest framework for getting a spatial map of transcription factor activity, which I'll go over in the next slide. And also what's going to help with the analysis is, since each of these spots does contain a multitude of cell types, there's cell type decomposition approaches, which can take our single cell data, the expected gene expression of each cell type, and take a good estimate as to the proportion of cell types within each spot. So then with that, we can try and relate the cell type activity, the cell type proportions to the transcription factor activities, and see if that relationship depends on any spatial context. If it's different in different regions of the sample, or the transcription factor is pretty much only dependent on the cell type proportions throughout the entire sample. That's kind of going to help us answer questions around, is the spatial context meaningful for a specific transcription factor? Okay, so the mathematical details of this algorithm, which is called Sagan, spatially informed transcription factor activity networks. We have a matrix D of transcription factors and their target genes, a matrix Y, which is going to have the spot-specific gene expression levels. And then at each spot, we have these spatial coordinates. That image, there's a few different ways to kind of embed that image into a vector that could be comparable. It's pretty much just taking the RGB values and saying, are these two spots, do they look vaguely similar? It's not doing anything too intelligent with the morphology of it. And then great. And then what we learn is a matrix of transcription factors, a transcription factor activities at each spot. And so this looks like, kind of like a standard least squares problem. I'm sorry, ignore that laser pointer again, where we have a relationship between these transcription factor target gene matrix and the gene expression. We decompose the coefficients into two terms. One of them is going to have this spatial structure enforced onto it, and stuff going to try and make it spatially coherent. The other one is just going to have a normal L2 regularizer. The idea being, we want to enforce the spatial structure, but some transcription factors might not have that, and so we want to leave that free to be kind of noisy, be kind of spatially incoherent. And the way this kernel regularizer works is essentially just saying that if two spots are close together and they look kind of similar, the RGB values in the image are similar enough, and then they should have a pretty similar transcription factor activity. This kind of works as like a multitask learning approach, where instead of learning the transcription factor activity at each spot separately, kind of pooling together data when it's relevant. Okay, so from there, we can do kind of another regression framework on top of that to correlate cell type proportions with transcription factor activities. So initially, this kind of makes the assumption that throughout the entire sample, transcription factor activity only depends on cell type proportion, which is sometimes true. Sometimes that explains all the variance in transcription factor activity. But just as interesting as when it works, I think, is when it doesn't work. From there, the idea is to usually go into it with some type of biologically meaningful way of separating the sample into regions. This could be proximity to like tertiary lymphoid structure, something I'm going to talk about later. It could be regions annotated by a pathologist. But within each region, we could fit this model and we can see, does this transcription factor activity have a different score for different cell types between the two regions? And if so, that might hint that there are some spatial context relevant to those regions that are influencing the transcription factors activity level. The data that I'm using to kind of demonstrate this comes from 3 different publicly available data sets that are from breast cancer tissue. And we have cell type deconvolution done for each of them. There's a variety of different molecular subtypes, which I'm doing some stuff to analyze and see if there's any meaningful things we can pick up there, but I don't think I'll talk about it in this talk. So, right, we have the cell deconvolution, as I mentioned. This is vaguely what that looks like. I have it grouped into 7 broad categories. And for each one, it's just a proportion between 0 and 1 of how many of the cells in that spot do we expect to be epithelial cells. And yeah, I say epithelial cells, so that is the dominant cell type in most of these samples, in most spots. And we have this transcription factor target gene data set. And that's the HTF target databases. I tried a few. This is the one that worked best. Right, so with that, I ran the model, I did all these data. And starting with this column right here, the second one, these are the transcription factor activities that are learned in this model. And the one next to it is, context problem would look like? And the answer is pretty similar, but kind of more staticky, kind of harder to get any meaningful insights from, especially when we look at this first column here, which is some correlated cell types. That's how I kind of picked out these different transcription factors. Is right, with this, we're seeing more correlation between transcription factors activities and the relevant cell types. ESR1 we see is highly correlated with epithelial cells, which we expected. If we look at just the non-spatial term, that trend would not be quite as clear. And also, if we looked at the mRNA level, again, it wouldn't be as obvious And some of the transcription factors, a lot of them really are lowly abundant in the transcriptome, so it's hard to pick up much signal with just this very sparse data. Right, so I think that highlights, some of the advantages of using transcription factor activities from this framework for spatial transcriptomics data. Another really neat thing about this is it clusters the data much better than just using gene expression. I did like a standard, lighting clustering and UMAP, first using the inferred transcription factor activities and also using gene expression. I also did this for transcription factor activities without using spatial context. It looks more similar to the bottom one here. But in the top one, we see these clusters are much more separated, they're much more distinct, and they're also more spatially resolved. They correspond to spatial domains within the sample. As opposed to just looking at gene expression, even if we kind of pick out some highly variable genes, it ends up being much noisier. Okay, so, kind of a key advantage of having spatial transcriptomics data is we can show this, across the image to a pathologist and ask, how would you label each of these regions? And so that's what we have here is a few different regions highlighted. And just for the interest of time, I'm going to highlight a couple. So in the orange region here, that's invasive cancer and lymphocytes. And we see the IRF family coming up a lot, which is kind of interesting to see. It makes a lot of sense. That's often associated with the response to invasive cancer, the IRF family. And in the other normal stroma and lymphocyte region up here, right, we see a lot of canonical markers like AR and GATA3 associated with that. So that makes sense. Kind of the next question I want to answer with this is, about tertiary lymphoid structure. Because the kind of cool thing about spatial transcriptomics is we can say in the spot, are we seeing a combination of T cells and B cells, which is kind of a way to characterize this tertiary lymphoid structure. It's a bit more specific than that. But what's kind of interesting about this is when it forms in breast cancer patients, it's essentially positive clinical outcomes, and patients who have these tertiary lymphoid structures in proximity to the cancerous regions of the tumor, they're more likely to respond to immunotherapy. So it'd be kind of cool to interesting to understand the transcriptional regulators that might be driving tertiary lymphoid structure. And right, so I have there's a way to kind of score spots based off of how likely they are to have this tertiary lymphoid structure. That's from this publication down here. And what I do is separate the sample into two regions, this orange one here, which is in proximity to TLS, and this blue one here, which is not. And then we're going to see what's the differences in the relationships between cell types and transcription factors in those two regions, and when are they different. Okay, so this bottom graph here, this cluster map is showing for all the samples based off of whether or not we have TLS or not. This is actually dividing them into regions, but we're able to see a few different transcription factors that are more highly associated with lymphocytes in patients that do have this tertiary lymphoid structure. And then these box plots are up here are from, I talked about there's like dividing into these domains and fitting this cell type transcription factor activity model separately. And we see that in lymphocytes, these two transcription factors are more active when in proximity to tertiary lymphoid structure. Okay, so that sums it up. We have this biologically interpretable and spatially informed dimensionality reduction technique is really what it is by finding these transcription factor activities that you spot. Using the cell type composition, you can delineate between context, spatial specific variants of transcription factor activities, and also cell type specific variants. And we're able to use that to find some transcription factors that are affected by proximity to tertiary lymphoid structure, or maybe even driving in. It's kind of hard to tell from this data any causal relationship, but we're able to see associations. Right, so I want to thank my lab and my funding from the National Library of Medicine.
Great talk. So if I understood the math correctly, all the heavy lifting of the spatial analysis is in that kernel function.
Yeah.
And it has this regularization weight, which there's no supervision possible here. So if you tune it differently, things will look much more beautiful spatially, but we don't know where the right balance is.
Yeah, so, I mean, I go back and forth whether I think about this as a supervised or an unsupervised method. But ultimately, when thinking about hyperparameters, I think about it as a supervised method. We're saying what I do is cross-validation to pick the parameters and best predict held out genes. So I might fit this model with 4,000 genes, and then say, how well does those transcription factor activities predict the remaining 1,000 genes? And so I pick the hyperparameters that best do that. And that's how I kind of avoid overfitting and putting too much weight in the spatial term.
Okay, we can talk about it offline.
Yeah.
I didn't quite follow.
Very interesting work. Thank you. My question is about the deconvolution step. So one of the major challenges in using single-cell RNA-seq as a reference for deconvolving spatial transcriptomic like Visium is the domain shift that exists between these two types of data. In fact, we did some experiment with that, and a simple classifier, let's say random forest or a support vector machine, can with 100% accuracy distinguish between matching data of the same cell type coming from single cell and spatial transcriptomic, which causes confounding effect when you use them directly. So I was wondering, how did you account for that kind of domain shift that exists in that data?
The methods I'm using aren't accounting for that. I'm kind of interested to see. So with the spatial data, you don't really have kind of a ground truth for the individual cell types, do you?
So, well, you can just know that, the different cell types. You can have the single cell RNA-seq as well. You can train a classifier and then ask whether it can tell whether it comes from single cell domain or just kind of a binary classifier. And that's very, very accurate, like too accurate for our comfort, basically.
Yeah.
Is that something you have looked into?
No, I don't think so. I don't know. I mean, the cell type deconvolution isn't really my focus. I'm just kind of pulling methods that have already been developed.
It's something to look into, because there are some methods now that they start taking that into account.
Oh, interesting.
Thank you.
Thanks.
So I'm just curious about the transcription factor and target database that you're using. Seems like a lot of the analysis that you're doing relies on the reliability of that data set. I'm just curious if you can comment on whether it's a prediction-based method, or how are they doing to have that database.
The data set itself comes from a combination of, I want to say there's some ChIP-seq experiments involved, there's literature evidence, there's gene expression correlations that are all going into the decisions for that. Right, so that is kind of one fault of this model, is it thinks that as truth, that's a ground truth, and builds these based on it. So I toyed around with the idea of adjusting it, kind of freeing that up as a variable, so we could reweight them, but it's not something I've ever gotten to work well with this data.
Thank you.
Thanks.
Okay.